has its analogy with Earth cartography, where we've had more than a millennium to chart the single surface of the planet we live on. That has progressed over over millennia to increasing levels of understanding, which, great. Start from quaint maps to wonderful hardbound book atlases, and then were revolutionized by satellite imagery coupled with informatics technology that allowed me to take a high-level view of where I started and where I ended this trip, and then zoom in on my lab and this building with incredible accuracy and ease of navigation. For cerebral cortex, we've had a mere century of cortical cartography, starting with the classical anatomist Brodmann and others, leading into invaluable book atlases, the Taylor Atlas most notably. Then we are experiencing a revolution in imaging technologies that are transforming our ability to acquire, analyze, visualize data, and do progressively more serious data mining. But at this stage, I would honestly say we're at a level of understanding kind of like that of 17th century Earth cartographers, to put it in perspective. So I want to make some fairly broad remarks framing the issue in the context of what do we know and don't know about macaque cerebral cortex, partitioning it, understanding its connectivity, and translate that up to the human cerebral cortex. The starting point is that we have many different ways of parsing the cortex into what we estimate as its constituent subdivisions. They are, in general, divisible into analyses of function, architecture, connectivity, and topography. And what we're witnessing is the emergence of even more methods, such as the, I don't seem to have a mouse there, the fMRI analyses of activations and resting state correlations, as well as MR-based analyses of architecture and diffusion tractography to drive the analysis. But staying with the macaque for a few more moments, it's sobering to realize that our current level of understanding is highly fragmentary. We have many different partitioning schemes. These are just a few exemplars, and we have a few areas, a relatively modest fraction of the total, where there is general consensus of what the area is, even if we disagree on the names. We can estimate that there are about 100, give or take maybe 20, distinct cortical areas in the macaque. That's based on the assumption that the entire cortical sheet can be cleanly divided into distinct areas. That's a hypothesis that's not yet proven. If we look at the range in size of cortical areas, it's notable that there's 2 orders of magnitude between the giant V1 and the smallest of the orbitofrontal areas that have been reported. It's also very important to remind ourselves that individual variability, say in V1, is a factor of 2 to 3 from one person or one monkey to the next. Coming back to the connectivity issue, we have literally hundreds of pathways amongst dozens of areas, probably thousands overall when it's more fully charted. And the complexity goes beyond that, because for any given tracer injection that one can do in the macaque, one sees a very complex pattern of connectivity that transcends just a connection in one area or not. On average, there are about 10 major inputs to and outputs from each cortical area talking to other cortical areas, not to mention lots of connections with subcortical regions. These pathways are almost always reciprocal, which is important when we try to interpret DTI analyses. There's an enormous range in the strength of connections, at least a couple of orders of magnitude. And as I alluded to before, the connectivity within any one, associated with any one area is heterogeneous based on topographic maps and or modularity within that area. And lastly, it's important to be mindful that adjacent areas almost always have rather similar patterns of connections. You don't see dramatic changes from one place to its neighbors. Functional connectivity as assessed by resting state analyses is an increasingly important approach. You'll hear about it today. The basic point that I want to make from studies in the macaque monkey are twofold. One is that if one places a seed, as was done here in the area LIP, one sees a pattern of functional correlations that are strikingly similar to the anatomical connectivity pattern shown on the right from Tim Lewis's tracer injections in the same area. That's heartening, but it has to be coupled with the awareness that functional correlations definitely transcend direct anatomical connectivity. And the best evidence I'm aware of is from this Vincent et al. Study where we put a seed in peripheral V1 in one hemisphere, that's representing the contralateral horizontal meridian, and there are strong correlations, robust correlations, with the opposite hemisphere calcarine sulcus. There are no wires there. It takes at least 3 synapses to get from one side to the other, and in that sense, functional correlations are often polysynaptic.